So good evening everyone. I already introduced metabolic pathways this week and still on carbohydrate metabolism. We are stressing much on how important the cell is and its organelles or the subcellular components, because the reactions which we defined much when we're defining biochemistry in my introduction slides talks much about the reaction. We are trying to understand biochemistry as chemistry of life, but then as much as what the life entails is the chemical reactions that take place. And these chemical reactions are the metabolic pathways, which takes place in the subcellular components or the organelles. And that is what makes us living, because of the chemical processes which takes place in those biomolecular components or the biomolecules we've been talking about. So we've introduced the biomolecules, and now we are introducing the metabolic pathways which takes place in the organelles. So I already introduced glycolysis, breakdown of glucose to pyruvate, to the main objective of generating energy in form of ATP. I also introduced gluconeogenesis, which happens at the point when the glucose level is low. So the body synthesizes glucose from the non-carbohydrate precursors like amino acids, the glycerols and lactates. I've also introduced a session where we saw the amphibolic role of glycolysis. Still, in one of the medical and biological importance of glycolysis, we saw that it is the same pathway in which fructose and galactose is metabolized. So I introduced the slide how fructose and galactose enters the glycolytic pathway, how they provide energy. So today I'm still going to proceed with the amphibolic role of glycolytic pathway. Glycolysis is the most important pathway to learn about, being that it provides materials for other pathways, or it gives the precursors which are needed for other pathways. So today I'm going to dwell on the TCA cycle, which is also known as the Krebs cycle. The other name is the citric acid cycle. And if you look at the TCA cycle and embarking much on glycolysis, I talked about the fate of pyruvate, and I mentioned that one of the fate of pyruvate in aerobic states or in aerobic condition, meaning in presence of oxygen, it was converting our pyruvate to acetyl-CoA. So the acetyl-CoA, very important for entry into the TCA cycle. We need acetyl-CoA, and that acetyl-CoA will come from glycolysis. So after forming pyruvate in aerobic state, we'll be able to form acetyl-CoA. So the overall goal for the tricarboxylic acid cycle is to make ATPs. It means the GTPs, the FADH2s, the NADH, which are going to be produced by this pathway, can be converted to ATP. It's also going to generate the NADHs and the FADH2s, which are going to be used as reducing equivalences. So they are reducing equivalences which can be used as electron carriers in electron transport chain. That one we are talking much about the oxidative phosphorylation and the electron transport chain, which is also a source of ATP production. So the pathway for the TCA cycle occurs in the mitochondrial matrix. We talked about the subcellular components, the organelles, involved much in hosting these chemical reactions. So this one takes place in the mitochondrial matrix. Remember, we already saw The glycolysis was taking place in the cytosol, and then for the gluconeogenesis, there are some which were taking place in the cytosol, some in the matrix, some in the endoplasmic reticulum, and we mentioned specific enzymes for the bypassed steps where each of the reactions were taking place. So this one takes place in the mitochondria, and specifically the mitochondrial matrix. So our acetyl-CoA, we have to convert it from pyruvate, but this involves a complex of 3 enzymes. So we have a multi-enzyme complex, meaning it contains 3 enzymes for it to be converted into acetyl-CoA to allow entry into the TCA cycle. So the first enzyme is pyruvate dehydrogenase, which has a component of the vitamin thiamine surrounded by 2 of the phosphates, the pyrophosphate. So it's also needed there. You have the dihydrolipoyl transacetylase, which has a CoA as a substrate. You can see a CoSH as a component there. That is one of the enzyme, dihydrolipoyl transacetylase, carrying it. And then we have dihydrolipoyl. We have another enzyme as well, dihydrolipoyl dehydrogenase, which is also a component there. So we have the 3 enzymes which are involved in the conversion of pyruvate to acetyl-CoA. But the acetyl-CoA, which is converted from pyruvate in aerobic state, has different options. It can go to the TCA cycle to generate the reducing equivalences as well as ATP. It can also go to the formation of triacyl glycerols, or it can go to formation of ketone bodies. So it has many pathways where it can go. But in this case, we need it for the Krebs cycle or the TCA cycle for the synthesis of the reducing equivalences, NADH and FADH2, together with the synthesis of ATP. So the acetyl-CoA is here. It has been converted through the complex of enzymes, the 3 enzymes which are involved in the conversion. The acetyl-CoA will combine with oxaloacetate. So it has to find oxaloacetate available for the TCA cycle. If the oxaloacetate is not available for the acetyl-CoA to combine to form citrate, then it will go to the lipids formation or lipid metabolism to form ketone bodies. So in absence of oxaloacetate, the acetyl-CoA will have access in ketone bodies formation, and it will be used in the formation of ketone bodies. So it has to find enough of the oxaloacetate, then it will combine. Acetyl-CoA will combine with oxaloacetate to form citrate. And the enzyme is citrate synthase. We have water going inside, and we have to remove the CoSH, which is on the acetyl-CoA. The enzyme transacetylase, which had a component of CoSH, now it has done its work. We'll remove the CoSH. The citrate synthase will remove the CoSH. We'll add water, will form citrate. So oxaloacetate is 4 carbons. Acetyl-CoA is 2 carbons. Our citrate will be 6 carbons. 1, 2, 3, 4, 5, 6. We are forming citrate. The enzyme is citrate synthase. First step, the enzyme is citrate synthase, forming citrate. The Krebs cycle has 8 steps. Step number 1, the enzyme involves citrate synthase. We are synthesizing citrate. Then citrate is converted to isocitrate. In this case, it is an isomerization. So the aconites will convert citrate to isocitrate, and in this case the aconitase, the enzyme, catalyzes this step. Then step number 3 is the first step where the first reducing equivalence NADH is forming. The enzyme is NAD dependent. It goes in depending on NAD. We are generating NADH, the first reducing equivalence. The same step, there is decarboxylation, removal of one carbon. So it means the resulting product, alpha ketoglutarate, is 5 carbons because we'll be having 6. Decarboxylation takes place here. The enzyme which catalyzes this step is isocitrate dehydrogenase, step 3. The first enzyme where NADH is generated is catalyzed by isocitrate dehydrogenase. Decarboxylation takes place in that step. We generate NADH, the first reducing equivalence. Alpha ketoglutarate is converted to succinyl-CoA, the second step where NADH is generated, another decarboxylation taking place, but now we are adding a CoSH group. So the resultant succinyl-CoA has a CoSH group up here, as well as it is reduced 1 carbon less, so it is 4 carbon atoms. The second step where NADH is generated is catalyzed by alpha ketoglutarate dehydrogenase. Then after forming succinyl-CoA, we generate GTP. Remember, it can also be equated to ATP. That was the main objective of this pathway, to generate energy in form of ATP. So this enzyme is GDP dependent. It releases GTP. We also remove the CoSH to form succinate. So just by removal of CoSH group, and we have a carboxyl group here, we form succinate. The other carbons remain intact. The enzyme is succinyl-CoA synthetase. Then from succinate to fumarate, you can see a double bond is forming from succinate, which didn't have a double bond. The enzyme is FAD dependent. We are generating a reducing equivalence FADH2. This enzyme is unique. It is the only one, the TCS cycle, which is FAD dependent. That is succinate dehydrogenase. So we are forming a fumarate, which is now a double bond. If you look at the class of enzymes, this one falls under the lyases. A double bond is going to be forming here. So after formation of fumarate, addition of water, we are going to form malate. Then the last step where the reducing equivalence is formed, the third reducing equivalence, is malate dehydrogenase, catalyzing the step. Malate is converted to oxaloacetate. NADH is generated, NAD dependent enzyme. So the cycle goes on and on. There has to be another available acetyl-CoA. It will combine with oxaloacetate. It will form citrate. So this is a type of cyclic metabolic pathway. Glycolysis and gluconeogenesis were linear type of metabolic pathway. So for the Krebs cycle, or the TCS cycle, or the citric acid cycle, the main objective was to generate the reducing equivalences NADH and FADH2, as well as to generate ATP. But still, the carbon dioxide which are generated here, can be used as carbonic acids whenever we are synthesizing the carbamoyl phosphate in the urea cycle. If we need the entry point where the carbamoyl phosphate has to be synthesized, the source of the carbon dioxide together with the amine group from the amino terminal end of the amino acid after digestion and degradation of amino acid will give us the carbamoyl phosphate, which is also the entry point for urea cycle. So we've generated the reducing equivalences. At the step where the dehydrogenases are, they are classified as oxidoreductases. So we had the first isocitrate dehydrogenase, the first enzyme involved in generation of NADH, alpha ketoglutarate dehydrogenase generating NADH, then malate dehydrogenase generating NADH. The enzyme which would only generate FADH2 was the succinate dehydrogenase, which is an FAD dependent enzyme, while the other three, malate dehydrogenase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase, are NAD dependent.